Good morning, everybody. My name is Alan Schroeder, and I'm the chair of the Pediatric Grand Rounds Committee. Welcoming today, Dr. Leonard unfortunately had another obligation, so he's not able to be here with us. But we have a wonderful talk today that Dr. Bonnie Maldonado will introduce very shortly. And then in the meantime, just some quick housekeeping. The text code for CME credit will be put into the chat. We have two really exciting grand rounds coming up. Next week, we have our own Paul Wise. For those that aren't aware, Paul has been working as a special expert in the U.S. federal court that oversees the treatment of migrant children in U.S. detention. We'll be sharing his experience for that next week, which is of course very important. And then Rudine DiCarlo. Rudine was a former pediatric resident here back in the 90s. He passed away during residency, and we resurrected this lecture series a couple of years ago. And I have an incredible speaker coming from Emory. Dr. Manning is the associate chair for GE&I there, and is really a prolific writer and fantastic speaker. So two great talks coming up. Next slide, Ingrid. Our next, actually really our last COVID in Children seminar series, we'll take a break during the summer, but next week we're gonna have another open forum and bring in a couple of general pediatricians to talk about how the vaccine efforts are going in primary care clinics around the county. Dr. Floyd and Dr. Jabati will speak on that, and then Dr. Lee will be here to answer your many questions about vaccination and all of the things related to COVID in children. Next slide. And then Children's Week, really great lineup of 5 different talks from June 14th to June 18th from various faculty here at Packard. And Ingrid will be putting the link for the COVID in Children series. She'll put a link about how to pre-submit questions in the chat, and we'll also put a link for this in the chat as well. And with that, Dr. Maldonado.
Thank you, and good morning, everybody. It's really a pleasure to introduce Dr. Blish. I've known her, it's been 10 years now, and I've watched her really become an international leader in her field. Dr. Blish is an adult, I wish you were a pediatrician, but adult infectious disease immunologist and stellar now COVID researcher. She went to UC Davis as an undergraduate and then got her MD and her PhD at the University of Washington, where she continued on as a resident and a fellow. We were fortunate enough to have her come to Stanford 10 years ago now from the University of Washington, where she was a faculty member. She is the co-director now of our MD PhD program here at Stanford School of Medicine. She's published internationally recognized work around the area of immune correlates of infection, malaria, HIV, many other diseases, but more recently, she's been working and really almost single-handedly developed our COVID-19 BSL-3 laboratory facility. It's one of the only ones, I would say, in California, and one of the top programs in the country at this point, given how rapidly this virus has evolved. She's just done an amazing job at building out our capacity here at Stanford to do some of this integral work around COVID pathogenesis. So we're very excited to have Catherine here at Stanford. She's one of these people that is always cool and calm, and I'm sure is working 25 hours a day and has done a wonderful job of pulling the COVID research together and helping us understand this virus. So today she is going to talk about COVID-19, and look forward to hearing what she's going to tell us our next steps. So thank you so much, Catherine, for being here, and please go ahead.
Wonderful. Well, thank you so much, Bonnie, for that really kind introduction. It's been a pleasure to be here. So first, thank you. It's really an honor to have the chance to talk to you today. And I am not a pediatrician, so I'm going to talk mostly about some of the more basic and translational aspects of our research into COVID-19. But I wanted to begin with a little bit of a framework into how my lab has approached our studies of infectious diseases. And pictured here along the top are 5 of our favorite pathogens that we're still currently working with. And for those who aren't familiar with what cartoons of bugs look like, we have HIV, influenza, dengue virus, tuberculosis, and of course, SARS CoV-2. And really, the approach that we've taken for many years in my lab, which has ended up positioning us well to tackle SARS CoV-2, has been, sorry, let me get everything, is to really understand what these pathogens know. And by that, I would argue that these pathogens actually know our immune system better than we know it ourselves, because by definition, in order to be a pathogen, each one of these pathogens has figured out a chink in our protective armor that is our immune system. So it has figured out a way to get past our defenses and actually cause disease. And the goal of my research program is really to take these different pathogens and understand which aspects of the immune response can stop a pathogen in its tracks, and which ones may be flaws that we can then bolster through immunotherapies or other issues. So with that framework in mind, we take a tripartite approach to tackling these questions across various pathogens, which is really centered in immune profiling of clinical cohorts, because we work primarily in human studies, which of course has its advantages and disadvantages. The beauty of immune profiling is, as shown in this cartoon here, we can, for instance, look and say what's different in severe disease A versus healthy controls. But you can't really establish mechanism with such studies because they're going to be mostly hypothesis generating. In order to do that, we've spent a lot of time building in vitro model systems for mechanistic studies, an example which I'm shown here with a lung slice infection that I'll talk about. And increasingly, we're also working to develop new methods to engineer immune cells, both to fight pathogens and also to feed into these mechanistic studies in order to prove whether certain pathways are important for a certain disease. And of course, our hope is that in the long run, we won't just use these for science, but actually be able to develop immunotherapeutics, for instance, for things like treating the latent HIV reservoir and contributing to HIV cure pathogens. So I'm not going to talk much about the engineering, but I did want to mention something very quickly, which was we have worked very closely with Paul Wender and his graduate student, Nancy Benner, in the Department of Chemistry, alongside my graduate student, Erin Wilk, in optimizing a method that Nancy and Paul really developed using so-called charge-altering releasable transporters. And the beautiful thing about this is that these carts allow us to really carefully and easily transfect cells with RNA. And one nice thing about this has been that it's been generally relatively straightforward to transfect adaptive immune cells, T and B cells, but tackling innate immune cells has been really challenging in terms of exploring their function. And I'm just showing here some work that Erin and Nancy did to transfect a variety of cells, including T cells. This is not a T cell optimized cart. We have ones that can be much better that he's done in work with other individuals, but we can transfect neutrophils, NK cells, monocytes, and B cells. And this, again, will allow us to deeply explore the function of different molecules with this. But that's all I'm going to say about the engineering at this point. And instead, I'm going to move on to immune profiling. And in the frequency of this, I don't think more than a year into this pandemic, I need to tell anyone that understanding COVID-19 pathogenesis is important, but just to provide a little framework as to how we presented these studies. One of the first things we noticed, and particularly with the data coming out of China, was the highly variable disease course. And I think there's maybe a bit of a fallacy that this is unique to SARS CoV-2, but I want to emphasize, and as pediatricians, you probably all know this, that this is not unique. In fact, many respiratory illnesses take this course, but it does provide an opportunity to understand what determinants of severe disease versus mild disease are. And we knew again early on that severe COVID-19 was associated with a so-called cytokine storm, with induction of cytokines like IL-6, TNF that are pro-inflammatory cytokines, and probably coordinate induction of IL-10, an immunosuppressive cytokine. Now, we can argue over whether cytokine storm is the proper term for this, which I might touch on slightly later. But regardless, it's important to consider that these cytokines actually play important roles in inflammatory and immune responses. And at certain time courses in an infection, they're perfectly appropriate. But it became clear pretty early that at some point this is crossing over into dysregulation. And that is when severe disease occurs. And at some point, these are becoming directly deleterious to lung tissue. And we really wanted to understand exactly what happened and how that occurs. And just to give a somewhat gross picture of how this works, this is an image that we lifted just from the New York Times. And it's the lung of an unfortunate, relatively young woman, I believe she was 38 years old, who underwent lung transplantation due to severe COVID-19 pneumonia. And you can see that her lung is completely scarred, it's fibrotic, it's bleeding, and it has an absolute lack of airspace disease. And so clearly, this is the pathognomonic for inflammatory responses gone wrong. So with this in mind, we really wanted to begin to evaluate both severe cases to inform immunotherapeutics, try to figure out how we could block such pathogenic inflammation, but also mild cases to inform vaccine and prophylactic design, as well as to see if we could figure out how to turn a bad immune response into a good one. So just to restate the goal more clearly, our goal really was to elucidate the immune pathways that might lead to protective immunity on one side or immunopathology on the other side. And unsurprisingly, as I already introduced the tripartite approach, so far what we've been focused on are in vitro studies of viral pathogenesis involving lung tissue and organoid infections, as well as in vivo studies to assess immune function across a range of disease. And I'll try to touch briefly on these issues going forward. So with immune profiling of COVID, I'm just going to talk very briefly about some of the early work, in part because it's already been published and was published actually last June and accepted in May as the single cell atlas of the peripheral immune response in patients with severe COVID-19. And this was a really impressive collaborative project between two graduate students in my group, Erin Wilk and Nancy Zhao, leading the charge on the single cell sequencing, Arjun Rustagi, my fellow, who contributed to the analysis and the writing, and also along with Jonacell, who was Angela Rogers, my pulmonary critical care colleagues, clinical care coordinator. We're running around coordinating enrollment and collection of blood from patients. Phil Grant and Aruna Subramanian, two of my ID colleagues, helped us to identify patients. And this is a picture of my entire lab in obviously pre-pandemic times. And everyone on here is pictured because every one of these individuals gave up their lives in March under shelter in place to process blood for these the COVID-19 Biobank, which I'll touch on again later, because the CTRU was shut under shelter in place and individuals were somewhat worried about working with these samples. So they worked in 10-hour shifts covering 20 hours a day for from the period of March through July, processing the blood from these patients so that we could do such studies. And so really a huge shout out to my amazing lab. Now, of course, we are far from the only people who have profiled the immune response. This is just an example of papers that came out within a few months of ours. And I'll particularly highlight some great work by Bali Plandren, our colleague. And also I'll come back to this really amazing paper by a German group that was much larger than ours that confirmed and extended some of our findings by Schulte-Schrepping et al. But what we actually did was we were isolating PBMCs, so peripheral blood mononuclear cells, from COVID-19 patients who were inpatient at Stanford Hospital. And in a little bit of a whim, we decided to perform Seq-Well, a single cell RNA sequencing pathway, kind of on the fly as we were processing these samples. So while two of my lab members would be processing the blood, getting them ready to freeze, another two lab members, namely Nancy and Erin, would take some of the blood samples on the side and in real time run the single cell RNA sequencing as patients came in, and then we would sequence them. We did standard analyses using standard techniques, happy to talk about it more, and visualize them at this point with what was referred to as SROT version 3. So these were the first samples we looked at for that initial atlas. And I'll highlight a few things. This was a very small study, but anyone who's done single cell RNA sequencing knows it was actually large for a single cell RNA sequencing study at the time. We actually had 7 donors and 8 samples. One individual we actually sampled twice. One unfortunate thing was we didn't actually have the demographics as we were pulling these samples. We were just taking 8 consecutive samples and processing them as they came in. And you can see that they all happen to be men. Ironically, the samples before and after were women. So that was a caveat of this study. We got luckier when it came to disease severity. If you look over here at the ventilated or ARDS category, it turned out that exactly half, 4 of the samples, were in acute respiratory distress syndrome at the time we collected the sample. The other 4 were not. These were very early samples in March. In fact, when I floated this idea of doing this, within 5 days, my team had it up and running and collected these samples. And so you can see that the only immunomodulator given at the time was arguably azithromycin in some of these subjects. There were no steroids given or remdesivir or other drugs at the time of blood collection. So what did we see? And this is just a snapshot picture of what we saw. And for those who aren't used to looking at UMAPs, I'll walk you through this a little bit. Each of these dots on this on these 2 projections represents a single cell from an individual. In the panel on the left, I've colored the individuals by patient ID, with the orange colors being COVID patients. And although I didn't mention it, we collected 6 healthy controls in parallel, and those are all the blue dots. The darker orange dots confer these patients were in ARDS or were sicker at the time of collection. On the right, you can see what the identification of each of these clusters of cells were. And so if you just focus in, for instance, here on the monocytes, you can see that there's almost no overlap between the blue dots and the orange dots, telling you that COVID monocytes from COVID-19 patients are completely different from monocytes in healthy patients, and they're totally reconfigured. And you can see changes too in the T cells. We can see induction of plasmablasts, the antibody secreting B cells, as we might expect, and so forth. If we break this down and we just look at the frequency of cells, now what I'm showing is the proportion of cells colored by donor. So the blue donors are, again, the healthy, the lighter colors, the less severely ill, and the darker colors, the more severely ill patients. We observed several important findings. And again, this was very early in the pandemic, but we saw depletion of multiple innate immune cell subsets. So these early responders were basically being lost from the peripheral blood. And this happened in our CD16 monocytes, our plasmacytoid dendritic cells, gamma delta T cells, and NK cells. And this, of course, raises the possibility, does this mean that these cells are actually going to the lung where the inflammation is, and that's why they're lost, or are they being somehow cleared? And you can't answer that here. We also saw, as I alluded to earlier, an induction of B cell plasmablasts. So we lose the naive B cells and we induce the plasmablasts, those cells that make antibodies. And of course, this didn't come as a surprise, but was good confirmation. And we see this more dramatically in individuals who are sicker. And that's because these individuals are trying to respond to the infection. And although I didn't highlight it, most of these individuals were about 9 to 14 days following onset of symptoms by the time they were hospitalized. And we expect a peak plasmablast response to be somewhere between 7 and 14 days. So this makes sense that they are sick and they're mounting an immune response to this disease. We also, in perhaps the weirdest thing I've ever seen in a study, saw this population of developing neutrophils, as we called them. And you can see this kind of linear projection here, which looks very weird because you're normally used to seeing globs when you look in a UMAP space. And we call them developing neutrophils because they look like immature neutrophils or on various stages of development, which I'll show you. Now, this is weird because for those who don't spend a lot of time spinning blood, when you process blood to make peripheral blood mononuclear cells, most of the immune cells in blood are neutrophils, about 70%, but they typically pellet with the red blood cells. So you actually throw them away. Despite doing this, we're seeing this production of these neutrophils, indicating that they're hypodense, meaning they're still allowing us to isolate them in the PBMC fraction. And our first thought was, well, you can see so-called low-density neutrophils in certain inflammatory conditions like lupus, and maybe that's them. But if you look over here, those are low-density neutrophils. This orange cluster here, these are totally unique. Oops, sorry, I didn't mean to do that. And so I'll talk a little bit more about that later. Next, I just want to talk briefly about some of the findings we made on the monocytes, because these are important early responders to infection, and they were the most reconfigured. And sorry. So if we look at the monocytes again, and we just cluster in on these, you can see that, again, there's no overlap between the healthy monocytes practically and the inflammatory monocytes. And if you look here, what we're showing is the downregulation in blue and upregulation in red of genes in the COVID patients compared to the average of all the healthy patients. And I don't know if anyone will actually be able to see it, but what you observe up here are a number of MHC class 2 genes, which is important in that it's showing that the COVID-19 monocytes have downregulated MHC class 2, indicating that they might communicate less well with CD4 T cells, and this could potentially be an escape mechanism. And that had been seen before in MERS and the original SARS, but this was the first report in SARS CoV-2. Oops, sorry. And then we also saw upregulation of several so-called interferon-stimulated genes. But what surprised us the most was how patchy this looks. And this was why we were glad that we had looked at so many patients. So for instance, if we just looked at this cluster here, we would have said lots of ISG upregulation, but if we'd looked at these guys here, we would have said less so. And it didn't fully track with the severity of disease. What was the biggest surprise, however, at the time was the limited expression of pro-inflammatory cytokine genes by peripheral leukocytes. So showing here, I'm showing anything blue is expressing transcripts for these various cytokines. And what you can see is there's really just not a lot of blue going on. I already told you that these patients were severely ill. We expect that they're in cytokine storm, meaning that they should be secreting lots of cytokines. And really, there's not much happening. And this tells you one of two things. Either the cytokines are being secreted at the post-transcriptional level, because this is just RNA, we're not looking at the protein, or that all the inflammation is happening in the lung. And in fact, if we look more broadly at just any cytokine upregulated in these individuals in the peripheral blood, this is the top 8 most secreted cytokines. And really the only one that's present in even just 6 of the 8 samples was IL-32 and NK cells. And for the most part, there's almost no inflammation in the peripheral blood. And again, this subsequently was reported by other individuals, indicating a bit of immune paralysis in the peripheral blood, likely because most of the activated cells are actually in the lung and that there's dysregulation of the immune response occurring. Now, it's important to note, again, as I alluded to, that this does not mean that cytokines aren't present. This is a subsequent study that we did in collaboration again with Angela Rogers and Jenny Wilson from Pulmonary Critical Care. A couple of postdocs in my lab, Laura and Ann Mott, helped with the analysis of this data. And what I'm showing here are 6 of the cytokines most classically associated with cytokine storm. And we're comparing in orange the non-ICU COVID patients, in pink COVID patients in the ICU, and in the blue are ARDS patients or sepsis patients from prior to the COVID-19 pandemic. These were collected as part of Angela's ICU Biobank. And what you can see is that there's a lot of pro-inflammatory cytokines present, but a huge range in spectrum. And it's not like COVID is special. It looks like other situations of ARDS. And obviously we need more subjects to be able to see if any of these subtle differences, for instance, in IL-18 hold up with larger numbers. But it's important to keep in mind that as we think about targeted therapeutics, we probably want to target the individuals with the higher levels of cytokines rather than those at the lower end of the spectrum. So if I go back and talk now a little more about these developing neutrophils, if we just break out this embedding and look at them a little more, this is why we called them developing neutrophils, because they appear to be a collection of neutrophils at various stages in development. What I'm showing here is a projection of something called RNA velocity. It's an informatics technique that uses the ratio of spliced to unspliced transcripts to infer a developmental trajectory. And you can see that it looks like the neutrophils are crossing along this pathway and becoming more mature as they go along. And in fact, if you look on the right, this is a projection that Aaron built, and you can see markers of plasmablasts, which they're next to, and I'm happy to talk about that later. But you can see that they express markers of stages of neutrophil development. They go through stages with primary, secondary, and tertiary granules, and it almost looks like you're stepping down this developmental pathway as you make your way along. So this really made us wonder, because one of the weirdest things we saw was that we're wondering if we're throwing away the most interesting cells when we make PBMCs and the neutrophils are going away. And in fact, if you look at this projection, it's weird the way there's this sort of abrupt cutoff. And we hypothesized that this might be the point at which the neutrophils become sufficiently mature that they pellet with the RBCs and we lose them. But the fact that we saw these cells only in the most severe cases really made us wonder that maybe we should study the whole process of neutrophil development and see what role it was playing in pathogenesis. Now, for anyone who's ever worked with neutrophils, it's really challenging. They're incredibly sensitive to manipulation, to the point that less so in mouse, but more in human, if you even touch your pipette tip to the bottom of the tube, you can cause a chain reaction degranulation where the neutrophils will explode themselves and kill all the other cells, and you end up with a gooey, sticky mess. As a result, or in addition, they can't be cryopreserved, so you can't freeze them and study them later, which is nice for batching and where you don't have to be on call to process samples. But we realized that by using the Seq-Well platform, we might be able to study them by single-cell RNA sequencing, because unlike 10x genomics and other single-cell sequencing techniques, there's no microfluidics and no sorting. And even just going through a microfluidic pathway can cause the NK cells to degranulate and get poor quality data. But Seq-Well, we actually just lay the cells gently over this multi-well plate, and we're hoping that we could settle out and get good quality neutrophils. So with that in mind, we decided to give it a shot with a healthy donor. And what I'm showing here is, again, one of these UMAP projections marking all the different types of cells from the peripheral blood mononuclear cells of a healthy donor. We then tried to purify purify neutrophils from this individual. And you can see that we got this beautiful cluster of neutrophils, which looks fantastic. We then decided, well, what about eosinophils, basophils, or other granulocytes? And if we just lysed the red blood cells and loaded this on, we could actually look at all the immune cells except the red blood cells, which are in the way, and evaluate this. And importantly, we got good quality data. In general, when most people have tried to get good quality single cell RNA sequencing data on neutrophils, there's been a very small number of transcripts per cells. And what I'm showing here is the number of unique molecular identifiers and the number of genes per cell. And you can see that while it's decreased, it's actually not that different from the PBMCs, which is consistent with what is known about the RNA content in these cells. And we've got fantastic quality data. So now we can profile neutrophils. And next, we really needed a better infrastructure to capture patients across the spectrum of disease. To get back to the point I said earlier, because we're seeing these only in the really ill patients. So what is different about these ill patients versus the healthy patients? And specifically, and I'm going to use this schematic for the next few slides, we have healthy individuals who don't have COVID. There's individuals who are mild and outpatient and doing fine, moderate, severe, and fatal. And the subjects we looked at before were primarily in the severe and fatal with one moderate, and we really wanted to add in these mild and get a sense for what were determinants of good and bad disease. And to do this, we formed the Stanford COVID-19 Biobank. And this is just the leadership team of this, with many individuals, including the sponsorship of our Dean of Research, Ruth O'Hara, but we've got Kari Nadeau, Sam Yang, Angela Rogers, myself, you and Ashley, Andra Baumkolms, and then Ruth. And what was developed was a system to collect samples upon entry of patients, either into the emergency department led by Sam or in the Crown Clinic led by Kari. We collected biospecimens at day 037 and ongoing as we're now transitioning into studying long COVID. And again, in the early days of the pandemic, my lab processed these, and now luckily with Ruth's leadership, they're being processed through the CTRU. We collected phenotypic data and we're able to do systems biology research. And I just want to make a brief mention of the number of people and the effort that goes into collecting these samples. I already showed the picture of my entire lab, but these are some of the other individuals. This is the team that worked in the emergency department to identify, consent these patients and get the samples collected. Once the individuals were inpatient, Angela and Jonasel were coordinating the efforts along with all our amazing nursing staff to collect these samples on the inpatients. This is the Crown Clinic team for the long-term follow-up, where we're able to bring in less ill patients as well as return visits at 30 and more days onward. And then now that we're transitioning to study these in the CTRU for the processing and got them out of my lab under Beck Osborne's leadership, these are some of the teams involved with that, more of the CTRU staff that are involved in a picture of their study. and the Biobank team to coordinate the distribution of patients. So with this incredible infrastructure, this is what we've been able to collect. And I'm sorry, folks, this is all in adults. Hopefully we'll have some pediatric samples that others have collected. But enrolled through the ED, we have 620 patients, and enrolled through the Crown Clinic, 211 patients, many thousands of aliquots. And we have about 12 labs that have received samples and requests. We have a biobank request form where we can try and match individuals to the samples they want to pull. So with that resource in mind, what did we do with them? And what we did was a little bit more painful because we needed the fresh samples to study the neutrophils. So these were still processed within my lab. But what we undertook was a multimodal profiling across the spectrum of disease. And this was a study that was again led by Erin Wilk, my talented MD PhD student, in collaboration with Maddie Lee, another grad student in my lab, and Ben Parks, a graduate student in Bay Way, a postdoc within Will Greenleaf's lab, who were instrumental in allowing us to expand to epigenomic profiling as well. So the workflow for this was that we took healthy controls in COVID patients across the entire spectrum of disease. We took the RBC lysed whole blood, as I alluded to, for single cell RNA sequencing, so we could study the granulocytes and neutrophils in addition to the rest of the PBMCs. We also performed a TAC-seq to look at epigenome, and we performed proteomic profiling with CyTOF and with a particular focus on natural killer cells, which were in pre-pandemic times a major focus of my lab research. And we now looked at 64 patients, and you can see here them colored by the disease severity, their age, and the days post-positive swab. Oops. And you can see that most of them were collected in the acute phase, a few in convalescent, and that we had a spectrum of severity across different ages. And I'm not going to be able to talk for the sake of time and not putting you to sleep about all the data. In particular, I'm not going to talk about the epigenomic profiling, although we have some fascinating profiling that potentially explains some of the dysregulation in the monocytes that I talked about earlier, as well as a potential role for NK cells in particular in severe and fatal disease. So this paper is in press at JX Med and is online at the bioRxiv, and I'm happy to take any questions. But I'm just going to tell a brief vignette about the single cell RNA sequencing of neutrophils, because at the time that was a somewhat unusual approach. And so if we now look at the same kind of UMAP colored by disease severity, what you see is that we now have this beautiful collection of neutrophils. And in fact, we also have the developing neutrophils, which we were a little worried it was only 4 patients, and now we've reconfirmed them in an additional 17 severe patients. And a few things really stood out to us just looking at this UMAP. The first was, again, here's healthy neutrophils, here's COVID neutrophils, and you can almost perfectly see that the neutrophils predict disease status with mild, moderate, severe, and fatal, which has 2 phenotypes, interestingly enough, clearly distinguished. We also see that these developing neutrophils that I talked about earlier are really enriched in fatal cases. If we look down here at the monocytes, I showed you earlier that the healthy and the severe patients were totally unique. What was most interesting was the fact that the mild patients had actually relatively normal looking monocytes, which implies either that these individuals didn't release this pro-inflammatory response and that was protective, or they've already cleared the disease and they're returning to homeostasis. But a major theme was that mild disease really looks very normal in most immune cell subsets except the neutrophils. And then we also found some NK cell changes, as I alluded to earlier, in severe and fatal disease, which I'm not going to talk about. So in looking at how black those developing neutrophils were, we actually wondered whether developing neutrophils were a predictor of death, unfortunately. And so we found the top 5 developing neutrophil genes. So these were 5 genes that distinguish neutrophils from all other cells, and applied these to another cohort from Overmeyer et al., who had released their data. And we were wondering whether this was an independent predictor of death in bulk RNA-seq data. And so what Aaron did was he built a score based on these genes, and he scored the patients in their sample, which was one of the few studies that had a significant number of, unfortunately, fatal cases. And you can definitely see an enrichment with the score being higher in the fatal cases. And in fact, you can calculate the AUC for this, and the developing neutrophil signature actually predicted death with an accuracy of about 81%. And what's important about this is it was actually a better predictor than the SOFA score, the critical care score that is often used to predict bad outcomes, indicating that this is really a specific signature that indicates a pathogenic immune response. Now, many questions remain, including whether these are just a correlate of bad outcomes or they're contributing to disease, and that work is ongoing in the lab. We also wanted to look more deeply at these neutrophil phenotypes as I looked at, and again, the neutrophils are shown up here, some of the canonical genes that we use to identify them. If we then recluster just on the neutrophils, you can see what I showed earlier, which is that we have healthy controls, mild, moderate, severe, and fatal disease completely distinguished from each other. And in fact, if we look at the induction of these interferon-stimulated genes, we see a stepwise induction as the disease gets more severe. Now, they're activated any way you look at them. Here is production of cytokines with more severe disease going up. So unlike the monocytes, these are not paralyzed in the peripheral blood. If we look at a phagocytosis score, how much they're eating things, they're also more inflamed. And if we look at a degranulation score, how much they're extruding themselves, they're also more inflamed. So this tells you that these are really hallmarks of this inflammation in COVID-19. And in fact, if you even apply other scores, such as those from sepsis and ARDS published in other studies, these individuals look like they're in sepsis or ARDS, despite the fact that this is a viral infection, and most of these cases were bacterial in these prior scores. So this is overall telling us just how inflammatory this is, and it is consistent with our data showing the massive inflammation at the protein level. So at this point, we became very interested in neutrophil netosis. And netosis is a process by which neutrophils undergo a very inflammatory cell death, which is thought to lead to hopefully clearance of bacterial infection, but it's probably not productive for viral infection. And a transcriptional marker of netosis is PADI4. And again, you can see this stepwise induction. And I alluded to earlier the Schulte-Schrepping study. And you can see that in that study, we also see induction of PADI4 in the neutrophils that they were able to profile by mimicking some of our techniques early on. And this really presented us with a question, which is whether we could block this pathogenic neutrophil activation. Multiple other studies around this time were stating that netosis was associated with bad outcomes, including particularly the coagulopathies that we were observing. And this intrigued us because we were talking with Corleone de Oliveris, a graduate student in Carolyn Bertozzi's lab, and he was coordinating with Aaron Wilk, again, my grad student. And the question was, based on the, could we turn off this pathogenic neutrophil activation? And this is where the sweet and the sour comes in. Siglec-9 is a lectin, so a sugar-like receptor on the surface of neutrophils. Importantly, it's an inhibitory receptor. So ligating Siglec-9 turns off neutrophils. But what we saw in the Schulte-Schrepping paper is that in severe disease, Siglec-9 is induced as part of this inflammatory process. And this led us to wonder whether we could inhibit this pathogenic activation. So the model here is that inflammation is inducing the neutrophils to undergo this netosis, which is causing inflammation, contributing to tissue damage, and contributing to this hyperinflammatory cascade. And could we use a Siglec-9 agonist to block this netosis pathway? And so Corleone had developed some Siglec-9 ligands, and they're kind of, I can't possibly understand the chemistry here, but they're lactosyl polypeptides that are conjugated to a Siglec-9 ligand with a membrane anchor. And the way it works is that when you express this, it'll anchor itself in the membrane, it will ligate Siglec-9 and inhibit neutrophil function. And there are a number of controls, including this lactosyl polypeptide without the Siglec-9 ligand. And what Corleone could show was that if he takes neutrophils from a healthy individual, they will undergo netosis at some spontaneous rate. So you can see here, here's untreated neutrophils, and anything that turns green is netosis. If he stimulates them with a TLR agonist, you can see increased netosis. And if he uses his ligand, you can see it's back to baseline level or lower. And that's just quantified here. Here's his Siglec 9 ligand. He's inhibiting netosis, and this is what the curve looks like. And in fact, it's actually somewhat protective over the spontaneous netosis. So several individuals had reported, several studies had reported that COVID-19 plasma was sufficient to induce netosis in healthy neutrophils. And in fact, we could recapitulate that with our own subjects. Shown here are the rate of netosis in healthy neutrophils that are incubated either with plasma from healthy individuals or plasma from COVID-19 patients. And you can see that if you have plasma from COVID-19 patients, you induce netosis. If you add the control lactosyl polypeptide, you don't do anything. But if you inhibit if you inhibit neutrophil function by agonizing Siglec 9, you can block this pathogenic activation. And so we're really hoping that this might be a potential therapeutic pathway that could potentially be used in severe cases. Now, unfortunately, this membrane-anchored design does not make for a good drug candidate, so we're exploring whether we can develop agonistic antibodies that can do the same function that might be better drug candidates. And we'll obviously need to explore these pathways in vivo models as well. But hopefully this provides a nice story of how we could go from a very basic discovery that we see this pathogenic neutrophil activation into a potential therapeutic pathway. Okay, so in my last few minutes, I want to move into talking a little bit about these in vitro model systems for mechanistic studies. And of course, and Bonnie alluded to this earlier, the hard part was that back in March, we were faced with the challenge of working with a novel, highly pathogenic respiratory virus and relatively limited facilities to do so. So this is a picture of Arjun Rustagi, my infectious disease fellow who has really instigated and led our SARS CoV-2 research program. And he is getting ready to enter the BioBubble, which was the facility that was built for Carolyn Bertozzi for tuberculosis research that she'd been kind enough to share use of with us for our TB research. And when the TB research programs were closed down under shelter in place on March 17th, we actually reopened the BioBubble 2 days later to do SARS CoV-2 research under Arjun's amazing leadership. And in fact, you can see here an example of a plaque assay that he's done where each of these clearings indicates where a viral particle has cleared this monolayer of cells. And he was able to very quickly start growing the virus in this BSL-3 facility. And this was the team of intrepid investigators that back in March were doing it. And actually not pictured here was Jeff Iveson, my grad student who helped out early on as well. Here's Arjun again and Joe Buomono. And they're very happy because this was the first ever plaque assay that was done in there and it worked. And this is Giovanni Martinez Colon, who was another talented postdoc in my lab who led our TB program until SARS hit and then transitioned over and has been helping the SARS CoV-2 research program. Now, as soon as people heard that we were growing the virus, we had just a small smattering of faculty reaching out with ideas for things that we should test and look at for pathogenic studies. And so we realized very quickly that we needed to expand our capacity because this is just a subset of the individuals who reached out to us. And so we were very fortunate that with support of the Dean's office, we were able to get a gift to build a new facility. And this is showing this facility under construction. And for scale, that BioBubble is entirely contained in this, behind this wall in the little tiny room there. And in a 2.5 month build, we have a brand spanking new BSL-3 facility. If anyone wants to get in there, there's a virus room and a tuberculosis room. And we have 16 current users from at least 8 different labs, I believe. And we're ready to go and open for business and hoping to build an animal BSL-3 facility as well. And just some more pictures. It's by far and away the nicest BSL-3 I've ever seen. It has windows. It's not hidden in a basement somewhere. It's over in the keck. And we have lots of fun things like a sorter in there. So now that we have this, the question is, what do we do with it? And I just wanted to provide that little bit of perspective. But one of the first collaborations we started was a really productive collaboration with Calvin Kuo's lab. And specifically, Arjun in my lab was working with a system developed originally by Amin Saluddin, who was a fellow in Calvin's lab, now actually has his own lab at the University of Chicago. And the project has really in this stage been run by Shannon Choi, a talented MD PhD student in collaboration with Arjun. And the idea here was to study SARS CoV-2 pathogenesis in organoids, and specifically in distal lung organoids, because the distal lung is where the pneumonia is happening and where we wanted to understand how the virus was inducing disease. And what his lab had done was come up with a system where we could start with peripheral human lung, grow up clonal distal organoids, and then differentiate them either towards more AT2-like cultures or even basal cell cultures, which are more heterogeneous in their basis. And just as an example, when you grow these little mini organoids, this is how they look, this so-called regular organoid. They're typically suspended in a matrigel and they grow in the apical surfaces out there. And this is what one looks like. So they're really cute, and Shannon takes beautiful pictures. So you can see a basal organoid and all the nuclei and some ploidin and secreted globin. The nice thing, though, is that in working with Manuela Mieva and others, they developed a method where we could turn these inside out. And this is actually important because if we think about the way a virus enters the cell, it's going to enter on the ciliated surface. That's the side that is presented to the cell. And this was also important because Shannon early on discovered that all the ACE2 expression, which is the receptor, is expressed on the apical surface. So if you add virus to one of these organoids, it's going to hit the basal surface where there's no receptor, whereas on this side is where the virus is. And in fact, Arjun was able to show that he could take these organoids and infect them and see an induction of both spliced and unspliced RNA in the mock versus the infected cultures. And in fact, he could even do plaque forming assays and show that these could produce intact virions coming out the other end. So what we set out to do was infect these apical out organoids, and I'm showing this here, just a picture looking at double-stranded RNA, which is really only present in the setting of viral replication from a RNA virus. And you can see a mock infection here and a infection here, and you can see all these infected cells. And this is now looking 96 hours later, specifically at the SARS CoV-2 nucleoprotein. And again, you can see robust infection of these apical out cultures. Now, if we infect different kinds of organoids, here's a so-called AT2 organoid. These are the alveolar stem cells, which were known in the early transcriptional studies to express the highest levels of ACE2. And you can see, again, robust infection throughout this culture. And if we looked at the basal organoids, which have both ciliated cells, see these pretty cilia here, as well as club cells, which are important for secreting surfactant to keep the lung elastic, you can again see infection. And to our surprise, we saw robust infection in many club cells, which was not a cell type that we would have hypothesized would be infected, but may explain some of the ARDS type phenomenon if we're inhibiting our ability to make that critical surfactant to keep the lungs elastic. Now, in the upper respiratory tract, there have been multiple reports that ciliated cells are infected. We have so far not really seen that in our distal lung organoids, but it's also important to know that many of those infections dissociated the organoid first, and these cilia are actively beating and may actually sweep the virus away before it can attach. We don't really understand that. But one really fascinating thing about these organoids is that because they're clonal, we can actually do this in a medium throughput fashion and try to seed organoids, for instance, in a 96-well plate and explore therapeutics and other options. Now, a downside of this system as it is now is that I'm an immunologist, of course, and these are epithelial only cultures. So we're not able to study how the immune response works to these or the inflammatory pathways. And there are two ways we're seeking to address that. One is that we've been working very closely with Shannon and Calvin on air-liquid interface organoids that introduce the immune cells into them, and these are working quite well and showing promising results. And the other is that we've been looking at lung slice cultures. So these are intact human lung slices, and this has been a really massive and fun collaboration with multiple laboratories, including Mark Krasnow, who really led the effort. in collaboration with Julia Salzman, Steve Quake, multiple individuals at the CZ Biohub, as well as our CT surgery and pediatric colleagues and so forth. And the idea is incredibly simple. We take donor lung either from donors that have been rejected for organ transplantation or healthy tissue from near a tumor resection site and make very small 300 micron slices. And then Argent takes these into the BSL-3 and infects them with SARS CoV-2, dissociates them into a single cell suspension, which is actually quite challenging in the BSL-3, which he can tell you all about. And then we perform single cell expression profiling by single cell RNA sequencing. Just see how I'm doing with time. And one of the first and most important things we noted was that we could identify a lot of lung cell types, more than 40 lung cell types in this atlas, and this included epithelial, endothelial, and immune cells. And also importantly, if I just color this by the number of SARS-CoV-2 reads, we see a lot of SARS-CoV-2 reads or RNA indicating infection or at least uptake of virus in these lung slice cultures. So this really led us to want to explore which cells were infected more deeply. And this is a very much in process evaluation, so forgive me. But if we look at just the number of viral reads on a logged axis relative to the number of cells by the number of viral reads, what we observe is about 1% of the cells in the culture actually have 100 or more viral RNA molecules detected. And it's hard to imagine that's just uptake. That is probably indicative of real viral replication in the cell. But we are continuing to explore that further. And then, of course, the question is, which are these cells that are heavily infected? And we saw, again, here we see our compartments, epithelial, endothelial, stromal, lymphoid, and myeloid. We see the expected AT2 cells corresponding with what we saw in the organoid cultures. But we also, to our surprise, saw many cell types that we didn't expect to see, including myofibroblasts, several subsets of CD8 T cells, and by far and away the winner were alveolar macrophages, with the most virus per cell and the most number of cells infected. And so we've spent a lot of time trying to figure out, is this really a legitimate infection? And in fact, working with Julia Salzman group and the Sicilian algorithm, we can identify very specific splice variants of the viral RNAs, indicating that these RNAs are actually being processed, and it's quite likely that this is a legitimate infection, though this is, again, ongoing analysis. And if we look again in the macrophages and non-macrophages, in addition to so-called known and canonical subgenomic viral RNAs, we can identify hundreds of novel RNA forms that we're further evaluating. But what's also interesting is that we can actually observe takeover of the host cell. If we now look at the viral fraction, so the frequency of viral RNAs relative to host RNAs, what we observe is, of course, the vast majority of cells don't have any virus, whereas as you walk up this so-called infection pseudotime, looking at this overtake, you can see cells where upwards of 90% of the transcripts are the virus, and we know that this can be done. Now, this is interesting in terms of ordering and understanding the viral replicative cycle, and it also gives us a chance to compare so-called uninfected bystander cells with extensively infected cells and understand more about the dynamics of this infection. And we're finding some really interesting data in our preliminary analyses of these. And of course, we're trying to explore further which cells are really infected versus just eating the virus. So I think that was a lot of information very quickly, but I'm just going to bring it back to where we started with what we're doing in terms of understanding the disease. Our goal here was really to build systems to better understand how this virus is causing disease, and we've done this with pairing immune profiling, with building these in vitro model systems. And what we're hoping now is to better understand with our model systems where we can interrupt these pathways that are associated with severe disease, explore, test drugs, test hypotheses, and develop new immunotherapeutics in the long run. And with that, I just want to thank a million collaborators, many of which may not have even made it on this. So my amazing lab, I do need to update this picture, but needless to say, it's been hard to get pictures, and I sort of talked about them as they go. The support of everyone in building out the BSL-3 and setting up these experiments, all our collaborators to collect these samples, of course, our study participants and our funding sources for doing this. And with that, I'd be happy to take any questions. I'll go back here.
Well, thank you so much, Catherine. That was really a whirlwind tour, and it's especially impressive because I still remember when we were talking about building the new renovated facility, and just the fact that you've been able to get this work done. And I really want to thank you and also your team for really working under very difficult conditions, especially at the beginning of the pandemic when people really weren't sure how safe it was to do this. And I know we took all the precautions we could, but it was still, unknown what could be done safely. So I do have some questions here for you. The first one is, what the question is whether you were able to run single cell RNA-seq on frozen samples, or did they have to be fresh samples?
So that's a great question. We can absolutely run single cell RNA sequencing on frozen samples. And in fact, that's how we've done most of our studies of other infections, including SIV, HIV, and so forth. The one thing that is challenging is you cannot perform the granulocyte studies. So the neutrophil, basophil, eosinophils, those won't survive cryopreservation. There's no good way to store fresh whole blood. And the other thing that's interesting is that we don't know the degree to which this weird developing neutrophil population is actually present in all viral infections because we've never looked at fresh blood before. We did it partly for speed and convenience. You don't need that many cells to go in a single cell RNA sequencing study. You need about 100,000. And to keep the cells happy, you typically want to store about 5 million per vial. We knew we were going to want to do it, and we didn't want to have to thaw a whole vial just to do single cell RNA sequencing, and we didn't have all the other assays up and running. That turned out to be important. So for instance, we looked through Bali's single cell data. He has hundreds of patients and all of his samples were cryopreserved, and you cannot find the developing neutrophils there. So those cells don't survive cryopreservation. And we've looked in several other data sets and they're not there. So the only way you can get them is to analyze fresh samples. And the German study, having read our initial study, decided to process fresh for the same reason. And that's why they're the only study, I forgot to include the slide, but they see the same cells only in their severe cases in another 200 patients. So it's definitely a finding. But some of Pravesh Khatri's work implies that this may be more consistent across all severe viral infections, including dengue and flu. Yeah. And just nobody's found it because nobody looked at fresh.
Wow, that's pretty fascinating. So an evolving signal that you could potentially track down as a common pathway then. So the second question then is, what is the mechanism for SARS-CoV? I assume it's 2. It says SARS-CoV-3, but that's a little scary. So let's just call the SARS-CoV-2 inducing the novel circulating neutrophil phenotype. Do patients have any clues to the findings based on simple CBCs with DIPs, for example?
It's a really good question. I don't think that we've seen anything on a CBC with diff that would tell us. And I should mention, in most patients, they're fairly rare phenotype, less than 1% of total circulating blood cells, although there are a couple patients where 30% of the circulating blood cells were these developing neutrophils. But so far, we haven't seen anything obvious. That said, Pravesh has done all this great work for gene signatures. It wouldn't be that hard to use that 5 gene signature as a predictor, but there are probably better predictors that would be more broad. For now, we think it's a marker of the so-called emergency granulopoiesis, which occurs in the settings of these severe infections where the bone marrow is actually ramped up. And we see, and it's described in more detail in our study, a very myeloid skewing and an induction of early hematopoietic precursors in the setting of severe disease. Probably as you're cycling through these cells quickly, the bone marrow is cranked, trying to produce more of them through any means necessary.
Although the inflammatory syndrome in children is rare, are you seeking biosamples for this group? And can you speculate why exposed human neonates are less likely to be infected? So
great questions. So certainly We ended up not looking at MIS-C because several other groups at Stanford were already doing so, including Katya, why am I drawing a blank on her name? And also Olivia
Anushka, yeah.
Martinez and so forth. We're leading that effort. So rather than doing a parallel effort, we're helping them look at their data for some of these signatures we're seeing in adults. But definitely, if people have samples, feel free to reach out and I can draw the connections and so forth. In terms of neonates, we don't really know the answer, but there've really only been scattered reports of infection in utero. Probably the virus can't get past the placenta. And in general, babies, get a lot of mom's antibodies. Antibodies are clearly very protective from this disease when we see how effective our vaccines are. So it may well be that as long as mom has mounted a good immune response that has protected baby, as long as baby's breastfeeding and so forth, probably that is the protection. It may also be that they're less inflammatory in general. And since a lot of the pathogenesis is the pro-inflammation, it may be just that they're developing, that this is a case where you really want to ignore the virus. And maybe they do a better job of that. So all speculation, I have no data for any of this, but these are my hypotheses.
Right. Well, thanks, Catherine. And also just want to say that Makeda Robinson, also a postdoc here, who was trained with Shri Tanav and is working in my lab also, is looking at the impact of the immune profiling as well in pediatric populations too. So any samples are welcome. So contact Catherine or me and we can see who can use those. So Catherine, listen, this has just been amazing. I can't believe how much work you've done on this in less than a year. And I look forward to additional information, especially around the nets and potential immunomodulators for this virus. So thanks to you and thanks to everybody for joining us this morning. And have a great Memorial Day weekend, everybody. Thank you.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.